Okay, next we will be looking at another kind of mutations known as a chromosomal aberration. So this is not mutations to the gene sequence itself. It is mutation to the chromosome structure or number. And remembering that chromosomes are made of DNA sequence, right? So changing the chromosome would still change the DNA sequence and therefore cause mutations or affects the protein production as well. Yeah. Okay, so both gene mutation and chromosomal aberration are mutations. Okay, just that there are different kinds of mutations. Right. So for the chromosomal aberrations, two types, right? Change in the new numbers of chromosomes, resulting in either aneuploidy or polyploidy, or change in the structure of the chromosome. And there are four different mechanisms, right? Duplication, deletion, inversion, and translocation. All right, so very quickly, right? Deletion of the chromosome, right, refers to a deletion of one portion of the chromosome, right? Not the whole chromosome. If whole chromosome is aneuploidy, right? Change in the number of chromosome. Right, so the structure in terms of deletion would be one portion of the chromosome removed, and you can imagine, right, then whatever genes that are found in this region would also be removed. Okay, duplication is the doubling of a portion of the chromosome, right? So the chromosome becomes longer, you have more gene copies. Inversion is when the chromosome is broken off, it is joined back to the chromosome at an inverted manner. So you can see, right, this in terms of the bending, one thick band and two more narrow bands here, which is actually inverted when it joins back to the chromosome. Right, so it may change the gene sequence. Okay, if the point where this portion breaks off is actually in the middle of a gene, for example, then when you invert it and join back, then that gene would be joined with some other sequence, right? Then it can cause a change in the gene sequence. Translocation is when one part of the chromosome is broken off and then moved to another part. So in this case, it's a mutual or bidirectional translocation. It can be a single direction translocation as well, right? So then this chromosome will be intact but added with another part of the green chromosome. Right? So translocation can be one direction or both direction. So these are the different chromosomal aberrations in terms of structure, right? And then in terms of numbers, you have aneuploidy or polyploidy. All right, so ploidy refers to the number of sets of chromosome. Right? For us humans, we are diploid. That's why in our karyotype, which shows the arrangement of the chromosomes, right? There are always two per chromosome, two sets of chromosome, right? So we are diploid, right? Haploid is when there's only one set. And for polyploidy first, right? For polyploidy, that means there are actually more than two sets. If the normal is two sets, then if there are 3 sets, 4 sets, then that's polyploidy. For aneuploidy is not the complete set that is changed, but just one chromosome or 2 chromosomes within the set. So if there's a deletion of one whole chromosome, then for a diploid, original diploid 2n, right, one less chromosome, so 2n minus 1. If one additional one, then 2n plus 1, and so on. Okay, and the result of aneuploidy first, right, is due to a mistake in either mitosis or meiosis. Right, so you will cover mitosis and meiosis process later in the term, right? So I'll leave it for those lectures to explain further how mistake in mitosis and meiosis would result in aneuploidy, right? So for now, you just know about the phenomenon known as aneuploidy, right? For those who are not so good with this topic in the secondary school, right? Or you have not actually studied bio in secondary school, then you can leave it till when we teach you mitosis and meiosis, and then you understand the process a bit better. All right, polyploidy is also a mistake in the either mitosis or meiosis process, but instead of just 1 or 2 chromosomes extra, is the whole set. That's extra. All right. Okay. So some examples here, right? This is the normal diploid 2 sets, right? Here I got 3 sets, here I got 4 sets. This one got the normal 2 sets, but with 1 extra chromosome, right? So 2n plus 1. This one is the normal 2 sets again, but with 1 less chromosome here. So 2n minus 1. Okay. All right. So now comparison between gene mutation and chromosomal aberration. Gene mutation is a change in the DNA sequence of a gene, right? Affects only one gene, right? So only one gene locus. Locus meaning the position of the gene in the chromosome, right? So only one position is affected, only one gene is affected. For the chromosomal aberration, it's a change in the structural number of chromosomes, and that would change a number of genes, right? Because one whole section of the chromosome is changed, right? If it is a structural aberration, that one whole section of chromosome may contain more than one gene. Of course, if you change the entire chromosome in terms of numbers, right, then the entire chromosome surely contains many genes. Right, mechanisms, right, addition, deletion, substitution for gene mutation. For the chromosomal aberration, in terms of structure, it's duplication, deletion, inversion, translocation. In terms of numbers, right, it's due to long disjunction resulting in aneuploidy or polyploidy, right? Okay, so the result is shown below lah, and then subsequently no more blanks already, so I'll just leave you to read it yourself, right? That will be the end of the video.